thank you, Umesh. Thank you for the invitation. It's an honor for me to participate in the centennial birthday of celebration of Gobind, one of the great scientists of the 20th century. As Parul said, I first met Gobind in 1962 when I joined his lab as a postdoctoral associate, and I had the privilege and good fortune of knowing him and working closely with him for a period of close to 50 years. This is year 50 was the year he passed away. At MIT, our offices were right across the corridor from each other. Our labs were adjacent, and we had joint lab meetings and seminars. I therefore got to know Gobind and his students and postdoctoral researchers very well, and I came to admire Gobind greatly as a mentor, scientist, colleague, and a friend. And so here is the slide that Parul has already shown of Gobind and I taken outside the biology building, about 20, almost 20 years ago. Gobind has contributed so much to science, and he had so much wisdom and ideas to impart that every day spent to Gobind for me was a day of inspiration. Now, what I'm going to do is to give you vignettes of Gobind as a scientist, as a human being, his early years and all that stuff in some order, I hope makes sense. But anyway, that's how I'm going to give my presentation. Gobind as a scientist. He was a pioneer and a visionary in science. Trained in chemistry, he focused on applying chemistry to solve important biological problems in biological problems, and thereby started the field of chemical biology. His work, as Parul has mentioned, led to the elucidation of the genetic code, one of the fundamental mysteries of life then, for which he was awarded a Nobel Prize. And this is shown, slide shows him being awarded the prize by the King of Sweden. And the next slide shows him with the 3 Nobel awardees in medicine or physiology, Gobind Khorana, Marshall Nirenberg, and Robert Hawley. Now, Gobind was not content with getting a Nobel Prize. He soon followed his work with chemical synthesis of a gene, an accomplishment considered to be a landmark in genetics. The gene that he synthesized was the gene for a tRNA or transfer RNA. And yet another crowning accomplishment that took gene synthesis a step further was the synthesis of another tRNA gene, along with all the DNA sequence elements necessary for its expression in a cell, and demonstration that the gene was fully functional in the cell. So those were some of the really major accomplishments of Gobind in the nucleic acid field. His work on development of synthesis of oligonucleotides and the technologies developed for the use of for synthesis of DNA have had such profound effects that the research done today would not have been possible. So he really opened up biology, research in biology in ways unimaginable then. Some examples include cloning, DNA sequencing, PCR amplification of DNA, DNA diagnostics, DNA mutagenesis, DNA chips, and yes, even mRNA vaccines, synthesized using synthetic DNA genes as template for mRNA synthesis. So let me just show you a couple of slides to make this point. This is the synthesis of a transfer RNA gene shown in 1970. This is the first report of a synthesis of a chemical synthesis of a tRNA gene. And the second slide, next slide shows how, the next slide shows how the gene was put together. So the gene was synthesized by really doing the following. At the top, you can see 5 different oligonucleotides, 1, 2, 3, 4, 5, being put together. The 5 chemically synthesized oligonucleotides are mixed. When you mix them, they come together with a gap in between wherever there's a gap. And then the gap is filled by using DNA ligase. So the idea was to chemically synthesize small oligonucleotides, put them together, they will come together through Watson-Crick base pairing, and then DNA ligase will join them. So if you do that, then you have the thing that is shown on the top, now fully connected, all the phosphodiester bonds are formed. Now leave it, please. And then that has things called complementary or sticky ends that will join it to the next set of DNAs, right? So DNA ligase joins them on the basis of sticky ends that brings the DNA elements together, and then the next one, and so on and so forth. So doing it in 3 steps, you complete the synthesis of DNA, tRNA genes, starting from very short oligonucleotides as small as 9 or 10 and as large as 20. So that is how his plan for gene synthesis was. And that is exactly the scheme that is used to clone in cloning. In cloning, you bring the DNA things together, a plasmid DNA cut with a restriction enzyme together with a DNA that you want to clone. And that's really, this was the basis of cloning. And the next thing that I would like to show is, yes, this is how, what Gobind said, I can't read this now, but we hope that these principles will permit studies of the punctuation marks in DNA by adding up appropriate deoxypolynucleotides at either end of the synthetic gene. Same principle could be used eventually to add the synthetic gene to other genomes. Here, Gobind is already talking about gene manipulations at a time when no gene had been isolated. He was really a visionary. Okay. Now, in the next slide, now having synthesized the gene, Gobind had now the next problem to be faced was how to amplify the gene, how to make more of the gene material so you can do biochemical experiments in the test tube, in this test tube. And so he came with the idea, published in the paper, one of the papers, the principles for, and he said, the principles for extensive synthesis of duplex tRNA genes which emerge from the present work are the following. The DNA duplex would be denatured to form single strands. The denaturation step would be carried out in the presence of a sufficiently large excess of the 2 appropriate primers. Upon cooling, one would hope to obtain 2 structures, one containing the template strand complex to the primer, and DNA polymerase will be added to complete the process of repair replication. 2 molecules of the original duplex should result. So this is the way, his idea of how he would amplify his tRNA gene from 1 molecule to 2 molecules, and then from 2 molecules to 4 molecules, 4 to 8, and so on. That is shown on the next slide as to exactly what happens. Yeah, you denature the DNA, the 2 strands come apart, you anneal it with the primer, and then you extend the annealed primer using DNA polymerase, and then you have 2 molecules of DNA. This is exactly what PCR is. This was published in 1971, 14 years before the technology for PCR was published, and Carrie Mullis, who published that technology, got the Nobel Prize. And there are many in the field who felt strongly that Gobind should have been also included in that Nobel Prize. So that was, this is his contribution in biology, is that he not only elucidated the genetic code and synthesized the chemical gene, but in the process, he developed so many technologies, methods for synthesizing oligonucleotides, without which there would be no biology now, these days. So that is really his contribution. With all his accomplishments, and at the pinnacle of his scientific career, Gobind decided to switch fields from working with nucleic acids to working on membrane proteins. And as Parol mentioned, Parol was among the early participants in the membrane, in his adventure into the field of membrane biology. And Marcia Rosner, who is going to be one of the speakers in this symposium today, was the other one. Marcia was one of the graduate students at that time. So in membranes also, he was a pioneer, working first with bacterioopsin, a protein that absorbs light and protons pump. So it's what's called a light-induced proton pump. He showed at very early, working just after a few years working on membranes, he showed that it was possible to refold a completely denatured protein, membrane protein, in the presence of synthetic lipids and the retinol chromophore to form an active form of the molecule. And it's a feat that was considered impossible by most people in the membrane field. This work opened up the field of membrane biology and allowed for the first time a systematic study of making mutants and studying the properties of the mutant proteins in the vitro by being able to reconstitute them. And this work also led to the identification of two key amino acids that's involved in proton translocation, or transport of membrane, transport of protons from one side of the membrane to another. So this was a phenomenal accomplishment. Having contributed so much to bacteriopsin biology, Gobind then switched to working on rhodopsins, somewhat similar to bacteriopsin, but very, very much more prevalent in our cells. In human cells contain 300 or more proteins called GPCRs, G protein coupled receptors. And Gobind, by starting similar work on rhodopsin, really opened up the field of GPCR biology. And GPCRs are not only very prevalent, they're also among the proteins that are most often the target of many of the pharmaceutical drugs that we use today. It's a very important protein. And Gobind completely really opened up the field of GPCR biology. Now, Gobind was also a visionary in many respects. His work on membrane proteins spanned chemistry, biology, and physics. And he collaborated with spectroscopists, biophysicists, and physicists. So he started doing interdisciplinary research long before the current trend of interdisciplinary research, long before. In 1968, he wrote about the possibility of gene manipulations, I think I've already mentioned before individual genes had been characterized from any organism. So he really was thinking way into the future. And in 1971, as we already discussed, in thinking about a need to amplify the synthetic gene, he proposed a series of steps similar to what we know as PCR. I think I'm repeating here, but that's important to say that he opened up the field so that people could think of PCRs. He actually said what PCR really was. Okay, some early years of Gobind, I think Parol has already mentioned that he came from a pretty low, humble background. His father was a lowly taxation clerk. And yet he was very interested in educating his children. And Gobind and his siblings took advantage of it. In a place that had no schools, Gobind eventually found his way to his high school in Multan, in the district of Multan. And after that, he got into the University of Lahore, in Punjab University in Lahore. And then there, actually, he was already beginning to do research, and he actually published 2 papers in Lahore Philosophical Society. And one of the papers is shown here. It's written by something, yes. It's actually 2 papers. He published 2 papers. And these 2 papers really brought him to the attention of authorities. He got a fellowship to go to England, and the place selected for him was Liverpool in England. So this is his Gobind scientific journey. Starting in Lahore, Punjab, to Liverpool for his PhD work. From Liverpool, as I will tell you, he went to Zurich. An interesting story about how he got to Zurich. They went to from Zurich, as Parul said, he spent 1 year there, came home to India, couldn't find a job, but fortunately was able to connect with a friend of his whom he met in Zurich, went to Cambridge in England, worked with Alexander Todd, a famous chemist who himself won the Nobel Prize in chemistry in the mid-50s on nucleic acids, nucleotides and nucleic acids. And from there, he got a job in British Columbia, Vancouver, was his first independent position, did phenomenal work there. And from with Vancouver, came to Madison, which is where I met him. I went to work for him, with him. And from Madison to Cambridge, which is where he spent the rest of his life. So that was his scientific journey. And here it basically shows what he did in things. And the times, British Columbia Research Council, Vancouver, 1952 to 60. And that is really where his work began. Okay. Now, all of this I show you here because there are certain times Gobind is a person who really, he came from humble background and yet became an icon in biology. One of the greatest scientists of the 20th century. And the reason for it is that Gobind was a person with an extraordinary drive. And he was focused, he was dedicated to what he was doing. And one of his philosophies was excellence in everything, wanted to be excellence in everything that he did, striving for excellence. And those were the characteristics that led to his success, phenomenal successes. So at the various stages, we go through various stages here. Now here we show picture of him. Next slide. Picture taken at Etihad at Etihad in Wisconsin. Is Wisconsin before? Yeah, another before that is days in Vancouver, right? And then he came to Wisconsin. Next slide. Okay, Wisconsin in the 60s. When it came to MIT in the 70s. Now, how did Gobind get to Etihad? When he was a graduate student, Gobind in Liverpool, most of the literature that he read was in German, because most of the chemical journals, well-known chemical journals, were published in German. So he was determined, absolutely determined, to do his postdoctoral work in a German laboratory. And he wanted to work with Vladimir Prelog at the ETH in Zurich, the Federal Institute of Technology in Zurich. And what did he do? Well, he went to Zurich with his PhD thesis in hand and approached Vladimir and walked into Prelog's lab and approached Vladimir Prelog and literally begged for a little space to work as a postdoctoral fellow. And Prelog said, well, you come in tomorrow. And so he went to the next day. And this is all in this slide. I won't read the slide for you. And next, it says here, next day, Professor Prelog, with his typically enthusiastic and encouraging smile, said yes. And at my excitement added that he was glad to see people happy. And Gobind says, looking back now over the years, I believe that spending a year at the ETH with Professor Prelog is probably the wisest thing I ever did in my life. Now, the other thing about this, his 1 year in ETH is that he could spend on, and he published 2 papers in German working on alkaloids, the next 2 slides. And one thing about this 1 year in ETH is that he could spend only 1 year because he was not paid by the Swiss lab. So he had to literally support himself with money that he had saved while he was a graduate student. So only 1 year spent, but the year spent had to have, again, a momentous conclusions. Is that momentous effects? Is that Gobind, in trying to really better himself in speaking German, understanding German, at nights he was at home. He would take journals home and translate German journals into English. And he was doing that routinely. And in the process of doing that, he came across chemical called the carbodiamides, which had been discovered in the 1870s or around 1870 or something like that, but had been left, because most organic chemists had not really used it much and basically ignored it. And Gobind, reading that journal and understanding it, translating it to English and understanding it, thought that this compound could come in useful because this compound had the property of activating carboxylic acids. So he thought it could also activate phosphoric acids, phosphates, and if you could do that, that could be really used for synthetic organic chemistry involving phosphate-containing materials. Yeah, so this is the compound that he eventually settled on, dicyclohexyl carbodiamide. And this compound would be useful for everything that he did in Vancouver and everything that he did in Wisconsin. This particular compound, which he came across while he was browsing through the literature, chemical literature in German, and translating them into English. Okay, so one of the things that came out of his sheer determination to spend time in a German lab. Okay, some of the things that he was able to do in Vancouver really was the following. He was able to synthesize ATP in massive amounts using this thing called DCC, dicyclohexyl carbodiamide. You take AMP, AMP is activated, and then you add pyrophosphate, and pyrophosphate is joined to AMP, and you make ATP. And so he made a whole series of these triphosphates. And then he also found out that if you take AMP and you take another compound containing phosphate, you can join the two into a pyrophosphate linkage. And many of the nucleotide coenzymes in biochemistry contain pyrophosphate groups. And so he synthesized, among other things, coenzyme A. He synthesized NAD, FAD, coenzyme A, and many other stuff. So that's another compound he synthesized. And the next slide shows other compounds he synthesized is something called cyclic GMP. Cyclic AMP at that time was known to be so-called second messenger. Could not be synthesized in large amounts, but he found a way of synthesizing cyclic AMP. And then he also synthesized cyclic GMP, which was not known at that time, but now happens to be another extremely important second messenger. So all these came through his chemical synthesis. He also synthesized something called phosphoribosol pyrophosphate, a compound, a coenzyme that's essential for a compound that's essential for synthesis of purines, pyrimidines, and amino acids like histidine and tryptophan. And then on the right, he synthesized same slide, please. Yeah, he synthesized short oligonucleotides. All of this was done while he was in Vancouver. All of this is done using this compound DCC, dicyclohexyl carbodiamide. And you can see that the result of this attempt to learn German and translating papers from German to English led to all this work. And so all this work, this was really the heydays of biochemistry. And these compounds were very important, not available in large amounts, and Gobind had found a way of synthesizing it. And so this caught the attention of really all of the well-known biochemists of the time, Arthur Kornberg, Paul Berg, Saul Roseman, and they literally flocked to his lab to learn all these synthetic methodologies that Gobind was developing. And so this really, next slide shows the connections that he made with people like Arthur Kornberg and Paul Berg, who was shown here at a meeting held in Gobind's honor in Vancouver in 1993. Arthur and Kornberg both went on to win the Nobel Prizes. Paul Berg for cloning, Arthur Kornberg for work on DNA polymerase, and here is shown Gobind there. So this this really led to long-lasting friendships between people, and Gobind visited Stanford often. In fact, went to work with in Arthur Kornberg's lab and learned to purify DNA polymerase, which would be essential for his work on gene synthesis, for his work on the genetic code. And work on Paul Berg's lab with students, and learn to purify, and Jerry Herbert's lab, learn to purify RNA polymerase, which would be used to convert the DNA to RNA in his work on the genetic code. So these friendships that lasted for a long time really became very important for collaboration, cooperation, and really learning the technologies, passing on the technology from one lab to another lab, and so on and so forth. Okay, the next slide shows now, this is in the later years now, Arthur and Gobind literally fooling around in Arthur's house. Just just as a party, I think Gobind was visiting, and Arthur's house, and then the Dale Kaiser in the picture not shown here is Dale Kaiser, Bob Lehman, and all the luminaries of the Stanford biochemistry department. Okay, so the next slide. Now this slide shows what I mean. Is Gobind had gone to Arthur's lab, purified DNA polymerase, and he brought it back to Madison. And then he had synthesized short oligonucleotides containing repeating sequences. And he was assaying for DNA polymerase and assaying the conversion of these short oligonucleotides into containing repeating sequences into long DNA molecules containing repeating sequences. This is the DNA that he used for his work on the genetic code. So Gobind himself participated in that work. Not only did he didn't get the Nobel Prize, not only because he was able to direct the work done by others in the lab, but he himself was participating in the lab. He was very much of an experimental biologist. Yeah. Now, Gobind was always trying to better himself. And I give you an example of how he did that. Early in his research career, he knew that he had some deficiencies. He knew that because of his background, he had to improve on these deficiencies. And part of the deficiency was that his verbal skills lacked improvement. To communicate science to an audience and clearly, he had to learn to speak clearly and slowly, and he had to learn to speak without an accent that we all have. And he felt that he had to improve on these. And so, as he told me, what he did was, his in England, it's a tradition that most people in the lab go out on a Friday night, go out to eat, go out to have a drink at the pub, and relax. In general, relax. And Gobind did not do that most of the time. And what he said was that he would go home to his apartment, stay home, and listen to BBC Third Program. And BBC Third Program, he wanted to learn how to enunciate, how to speak clearly, how to enunciate properly, so that he would not have the accent, and that how he could be understood clearly when he gave a seminar, gave a scientific presentation, and things like that. And actually, he became an outstanding speaker and scientific writer, and would often exhort some of his younger colleagues who came to join his lab to also speak clearly and speak slowly. And that was, that's something that he was very conscious of. He improved, he became an expert at it, and he wanted everybody else to also try to do the same at least. In spite of all his accomplishments and awards and accolades that he received, Gobind was, as Parul has mentioned, as an extremely humble and modest at all times. He was, however, modesty only takes you so far. He was extremely modest. He was, however, a strong believer in Otto Loewy's motto: we must be modest, except in our aims. So in science, Gobind aimed very high, almost extreme in his ambitions. But once he had decided on a problem to work on, he prepared well. He would go to labs, learn the techniques needed to switch to membranes, working on membranes and membrane proteins. He went to Ephraim Racker's lab and learned everything that he could on how to reconstitute proteins using synthetic lipids and things like that. And he brought it back to the lab. He would do that. He would prepare himself very well for the job at hand. And he would stay on with the problem. He persevered. He focused on what he was doing and did not want to get distracted in any way from it. So those were the kinds of philosophies that he had that really sustained him and helped him attain his goals which is massive goals, just difficult, you would choose difficult problems. And he often said that if you want to get far, you have to travel alone. That was his motto. That if you want to get far in research, you have to be the one to begin doing it and be the only one to do it, because most people will not want to handle difficult problems. This slide is an indication of Gobind's focus. Now, this slide was taken on the day that Gobind received the Nobel Prize for his work on the genetic code. Yeah. And there was a party in the lab. The other directors of the Enzyme Institute had put together a celebration. You can see people with glasses in their hand. And Gobind here is telling these two people, one is a faculty member in the biochemistry department, and one is one of the other co-directors of the Enzyme Institute, exactly what he's doing. He was not telling them about why he won the Nobel Prize. He was not telling them about the genetic code. What he was telling them here on the day he won the Nobel Prize and at the time of his celebration was how he was putting together synthetic oligonucleotides to synthesize a gene. And that slide basically shows DNAs just being drawn as straight lines, and how he was going to bring different sets of DNAs together and join them with DNA ligase to go from small synthetic oligonucleotides to longer DNA molecules, a gene. And that is how focused he is that's the example of how focused he is in his work. Genetic code was almost passé at that time, gone, finished, done. Now he was on to his next adventure, and that was the synthesis of a gene. So that's really the gist of Gobind and Gobind. Yeah. Now, Gobind as a mentor. Gobind was also a terrific mentor. He set high standards and could be demanding, but he was no more so of others than he was of himself. By working closely with members of his group on a one-on-one basis or with a group, he set examples that they could follow and learn from. So Gobind really taught people by setting examples that others could emulate and others could learn from. Associates, people who work with him often said that writing a paper with Gobind was a really amazing learning experience. He was always so careful and meticulous. Every paper went through multiple drafts. And he cared deeply about the training of students and postdoctoral people in his lab, and went out of his way to help them, support them, and took great delight in their successes. So this is the day that two of his students got their PhD degrees. They had just come back from the convocation, MIT convocation, and here's Gobind in his regalia along with his two students. And these two students, by the way, were so productive that each of them published pretty close to 15 or so papers as part of their PhD thesis. So you might think that, it's all work and no play, right? This is really arduous thing, but not so. There was also plenty of play. And Gobind and Esther, his wife, were excellent hosts. And, they would have Christmas parties at the house or other parties at the house, or there would be lab parties, and the lab would go out for a day into a state park and have, food to eat and games to play and things like that. This is now Shilda Sharma in the middle, had just defended his thesis, and this is a celebration after his defense. So that shows me and Gobind. He still had worked as joint students of Gobind and I, working on a project in my lab, but of interest to Gobind, bacteria opsin genes and messenger RNAs and things like that. And so this is a celebration. It shows me drinking a beer, but not sure. But that's the kind of thing that goes on. And the whole lab would join us afterwards, right? Then the next one is now a lab party somewhere in Wisconsin, 1963, shows our friend Dieter Sol cooking a burger on the grill. So this is the lab party in a state park, okay? And afterwards is a party at Gobind's house on Christmas Day. Shows several things now. On the top is shows Esther and Bim, his youngest son, and there's a turkey you can see. And the next one shows Gobind with a group of people, also part of the Christmas party. And then on the left is Rolf Lorman working in a lab. On his right is several of us going to what's called a Federation meeting. We used to go to meetings together. Federation meetings were the meeting to go at that time. And so we would go there. And that's taken at the airport, I think. There's T.M. Jacob, who was a faculty, came from IISc in Bangalore, worked with Gobind, went back to IISc, and he was there for many, many years until he passed away a few years ago. T.M. Jacob, Dieter Sol, me, and Rolf Lorman and his wife and baby, all going to the Federation meetings. And then at the bottom is the picture. This is Gobind, Esther, and his 3 children, Julia, Emily, and Bim, or Dave. Yeah, so this is all put together by Dieter Sol, this picture, a picture taken in 1963, and he sent it to me, or I came across it. Right. Okay, so next slide is, now, yeah, Gobind also delighted in playing games. He'll play croquet or so. This is in the cabin that he had in New Hampshire. The lab goes there for a day. My lab, my group, his group, they all go there for the day, and we have a lot of fun, lots to eat. We go for walks and things like that. And here, Gobind is in the red shirt, is playing volleyball. So here we are going for a hike in the mountains in New Hampshire. So there's lots of outdoor activities, other activities. Gobind loved to hike. He loved music. He loved solitude, which is when he really thought. To me, I talked about Gobind as a mentor. To me, Gobind was extremely friendly, kind, and generous at all times. Like a kind note written to me as an autograph. He wanted to give me his autograph. He chose a Nobel lecture, and it says, "To Tom, in admiration and friendship, Gobind." So that's the kind of thing Gobind did. Always notes like that. And then the next note, which is next slide, yes, it's like that Dieter Sol and I had co-edited a book called tRNA, published by American Society of Microbiology. And one day I come back to my office and I see this note Gobind had written. "Did you see the wonderful review of your book in Nature, Structural Biology?" Gobind, 7/20. He always put dates also. Yeah, so this was done, just when the review came out. I don't even remember when it was. I had not seen it. The review was good, and I was happy. But this is he did rejoiced in successes of people his colleagues. Right. So, okay. Now, Gobind has left a tremendous legacy, obviously. One of Gobind's earliest postdoctoral associates, Gordon Tenner, said on the occasion of his winning the Nobel Prize, "He showed us what excellence in science was, and we learned to recognize it." So this was really Gobind's legacy, that everybody who went through his lab learned from him that excellence, striving for excellence, not that yet, yeah. Striving for excellence, everything that you did. And since then, generations of pre and postdoctoral students have come and gone through his lab, made their mark, learned from him about really how to do research, and the need for creativity, dedication, hard work, focus, stringency, and all this other stuff, striving for excellence at all times. These are the things that he really hammered on. He's sure how to do it, and we learn to recognize. And many of these people who went through his lab are now accomplished scientists on their own merit and occupy positions of leadership at established universities and businesses throughout the world. And the 2 speakers after me, Dieter Sol and Marcel Rosner, are examples of that, pretty superb examples of that. So the scientific legacy of Gobind Khorana really lives on. Now, the last thing I would like to say is that another legacy of Gobind is the program of Khorana interns, supported by the government of India and universities in the United States. The program originally started at the University of Wisconsin involving Asim Ansari, has now broadened to include universities throughout the country, throughout the US, and allows undergraduate students from India to spend a summer doing research in the US labs. Govind was very happy about this program and took great delight on meeting the interns. And here it shows him meeting some of the interns at a meeting in Wisconsin held in 2009, 2 years before Govind passed away. And all these interns were there, and Govind could not be happier. And he was there. And in the next slide shows some of the interns. And next slide shows at the same meeting was Marshall Nirenberg. Marshall Nirenberg came to that meeting, who shared the Nobel Prize with Govind came to that meeting. And behind the two of them are some of the people who were involved in starting the program, Khorana program. Asim Ansari is there. One of the deans of the University of Wisconsin is there. And a gentleman named Sam Petroda is there. And others. Okay, so next slide is, this is now at the same, after that meeting, Julius Adler, a dear friend of his, is invited us to dinner at his house. And at the dinner, Julius has brought out a bottle of champagne. And this is the bottle of champagne that when Govind got the Nobel Prize, at that same day, that night, Julius has given a dinner at his house for us, Govind and us. And so a lot of champagne was drunk. And Julius had saved one of the bottles of champagne for a time when they would celebrate again. And so this is Julius invited us to his house for dinner again, the day 2009, 2 years before Govind passed away. And then here, Julius brought out this bottle of champagne that had been drunk. It's just empty bottle. This is not a real bottle, but empty bottle. But they had written something in that bottle. Now, Govind had many, many awards and accolades, I said. I'll just give you two examples. One of them is the one of the lecture halls in the biochemistry department in Wisconsin now is called Gobind Khorana Auditorium. This is the unveiling of that auditorium during the meeting, Khorana Auditorium. And then people in Wisconsin, in Vancouver, have set a park aside for Govind in his honor. This is in the fringes of the university, just outside the university, Khorana Park. And next to the Khorana Park is the Smith Park. Smith Park is named after Mike Smith. Mike Smith won the Nobel Prize, who was a student of Govind's, was a postdoc in Govind's lab, and Mike Smith won the Nobel Prize also. So Mike Smith Park is right next to the Gobind Khorana Park. So that's really what I have to say. I think, Gobind was a tremendous, tremendous, tremendous source of inspiration for me. And I'm sure a source of inspiration to many. And I thought that, the folks in India would like to know how much we think of Gobind.
Thank you, Tom. That was really wonderful. How did Gobind overcome the, kind of inhibitions that most of us have while we want to interact with people that we have not seen before, or, kind of questions that one asks, things like that? How do we overcome all of that?
I think Roger Beer, the person who he did his PhD with, from what I understand, was a really, really good mentor. And he took Gobind under his wing. And in fact, Gobind became a friend of Roger Beer's. And Beer invited Gobind to their house for dinner, even when he was a student. And I think he wanted to bring him out. And Gobind, in turn, really wanted to improve himself. Because communication, maybe you don't. I did, certainly. That when I went to England, I had an accent. I spoke very fast. This is typical. I
Yeah.
spoke very fast, and so that people really are having a hard time on trying to understand you, and so you learn to speak slowly. I appreciate it when Govind would tell people who came to give seminars or interview for postdoctoral positions and things like that, that I didn't get to think, and would tell them to please speak slowly, and things. And the losing the accent, we all have accent, we still have accents, uh, but at least you can make yourself heard, understood. If you speak slowly and you try to really learn how to do it properly.
There's a question here, which is, I think, probably a question from everyone, but it is, from Umakant Subudhi. Uh, how to select a strong scientific problem in view of Khorana?
In view of Quran, well,
Yeah,
I
like,
think,
how would he? Yeah,
well, I think, it's in our fields it is different. You think you have to know the literature a little bit, yeah.
yeah,
And so you think of the problems are important, that you think are important, that remain unsolved. And uh, in Govind's case, I think once he went through Vancouver, he had opportunities. Yeah. So you need opportunities, and I think maybe in India also, you certainly have opportunity now in India. So in terms of selecting a problem, you think the important things are the problem has to be broad, of broad interest, of fundamental interest. The problem can be challenging. Problem can be considered to be very difficult. Some people can say this is not practical and all that stuff. But if you have the determination, if you have the opportunity, if you have the facilities, you can handle it. Now, I mean, Govind did not have the facilities. There are stories of how when he went to Vancouver, he was there was one fraction collector in all of Vancouver for separating fractions in of nucleotides and oligonucleotides and all that stuff, all chemicals. The fraction collector was in a different place, different room. The UV spectrophotometer was in a different room. So Govind says that they would go and run from the fraction collector to the UV room, take the reading, and then meanwhile draw the graph, to draw the graph of the fractions versus some reading, UV reading or things like that. And then they kept on going up and down, up and down, up and down. So they had that kind of problem, but determinism, the willingness to do things, willingness to achieve your goals, working hard, really was very important for Govind. And so he got some money at the beginning, but not much. But once the work gets going, once people know that this is a lab where you can learn things, and this is a lab where you can do good research, people flock to your lab. And once that happens, then that is the beginning of the establishment of a lab. So yeah, problem selecting a problem is the the level of difficulty of the problem is it's your choice. You can either select something that is you think is practical, I'll do that, and then I'll see where that leads, and then go to the next one. And I think some people do that, and others have this grand vision of a big problem, and and not only have the vision but willingness to stick with it and persevere. And Govind was belonged to that category.
yeah. I think there are a lot of these days data-driven kind of questions that are being asked. Yeah. So
Yeah, I
there's a
mean,
yeah.
yeah, yeah. I mean, I have my own thoughts on data-driven research, but uh,
So there is. I will take one more question, Tom, because we need to move on. There is a
okay.
question here. Govind didn't visit India often to encourage researchers in person. What do you think was the reason?
I don't know. I mean, I know that Govind visited India at least twice. Once I know that he even went to Nepal, and he went to Nepal and met my parents, met my family, in fact. Uh, and then the next time I was with him, and when we came to the Indian International Congress of Genetics in 1983.
I
And
remember that.
I don't know. I don't remember. I don't know whether he came to the International Congress of Biochemistry. Uh, uh,
No, he didn't. I think that
no, he
was,
did.
yeah.
He did. Yeah, I was there. I was there. I was there. But he, I don't think he did. Why didn't he go back to India often? I think this Govind's family was uprooted, right, from Raipur. They had to move after partition, and maybe he, I don't know. But yeah, it's true that he didn't travel as much to India. But then he didn't like to travel much. He went only when he had to give a lecture or something like that. Awards and others he would rather not travel to. Uh,
Thanks a lot, Tom. We really enjoyed